Good morning, students. So today we will study about the HGP, that is Human Genome Project. In our last video, we completed the process of regulation of gene expression, and in 12th class our next topic is HGP, that is Human Genome Project. So this Human Genome Project, it started on October 1st, 1990 by some molecular biologist, and it came to an end in 2003, that is 13 years after its start. Now this HGP, it is aimed at to map the complete human genome. So this HGP, it is aimed at to map the complete human genome by determining the complete sequences of nucleotides, in DNA of all chromosomes. All chromosomes means that is 22 autosomes, and X and Y as sex chromosomes. So this human genome, it is aimed at to map the complete human genome, and this mapping of complete human genome, it is done by determining the complete sequences of nucleotides which are present in DNA of all chromosomes, that is 22 autosomes and X and Y as sex chromosomes. Now this Human Genome Project, it was a mega project. Why we are calling it as a mega project? There are three reasons to call HGP as a mega project. The first reason is that human genome contains near about 3 billion base pairs. 3 billion base pairs. And the cost of sequencing single base pair is near about $3. So the total cost of sequencing 3 billion base pair becomes near about $9 billion. Further, the information that is obtained from HGP, it has to be stored, and for storage of this information, which we obtained from HGP, it requires near about 3,300 books. So storage of information which is obtained from HGP requires 3,300 books, and each of these books contains 1,000 pages. 1,000 pages, and each page contains 1,000 words. Now the third reason for calling HGP as a mega project is that this HGP, it requires some high speed computational devices. So this HGP, it is also associated with a new branch, with the development of a new branch of science or in biology, that is known as bioinformatics. So these are the three reasons. That's why we are calling HGP as a mega project. The third reason is because this HGP, it requires some high speed computational devices. So this HGP, it is closely associated with the development of a new branch in biology, that is known as bioinformatics. Now we will discuss about the goals of HGP. So what are the aims of this HGP? So the first aim of HGP is to identify and determine the function of each and every gene which is present in human genome. So approximately human genome contains 20,000 to 25,000 genes. So the first aim of HGP is to identify and determine the function of each and every gene that is present in human genome. Now the second aim of HGP is to sequence 3 billion nucleotides present in human DNA. 3 billion nucleotides means 3 into 10 to power 9 base pairs or nucleotides present in human DNA. So the second aim is to sequence the 3 billion base pairs or nucleotides which are present in human DNA. Now the third aim of this HGP is to identify and point out the genes whose mutation causes genetic disorders in human beings. So third aim of this HGP was to identify and point out all the genes which have undergone the mutation, and these mutations resulted in the genetic disorders in case of human beings. Now the fourth aim of this HGP, because we have obtained so much of this information from this HGP, and all of these information they cannot be written, they cannot be in the written form. So the next aim of this HGP is to develop, or even you can say to store the information which is obtained from HGP in the form of databases. So the next aim of HGP is to store the information which is obtained from HGP in the form of databases. Now these databases have to be analyzed, and for this we have to develop the tools. So the fifth aim of HGP is to develop tools for data analysis. Now next aim of HGP is to transfer this information which is obtained from it to the other sectors such as industries or to some pharmaceuticals. So the next aim of HGP is to transfer information to other sectors. Other sector means to industries such as pharmaceuticals. Now the next aim of HGP is to address all possible ESLI. ESLI means ethical, social, legal issue which may arise from this project. So these are the 7 goals or aims of HGP. The last one is to address all possible ethical, social, legal issue that may arise from this project. So this HGP, it was started on 1st October 1990 and it came to an end in 2003. And the first chromosome, it was sequenced in 2006. Now come to the methodology of HGP. So this HGP used 2 approaches, or we have 2 methodologies to carry out this HGP. The first one is expressed sequence tags. Expressed sequence tags, and sometimes its full form is asked in the exam, that is ESTs. ESTs is the expressed sequence tags. So during expressed sequence tags, all genes which are expressed in the form of RNA in a cell are identified. So during this methodology, that is during express sequence tags or ESTs, all the genes in a cell which are expressed in the form of RNAs are identified. Now during second methodology, that is known as sequence annotation. So during sequence annotation, all the genome or the total genome present in a cell is sequenced. That means it includes the coding and non-coding regions both, and then the sequence are assigned with the different functions. So during sequence annotation, sequencing of total genome is carried out, and this sequencing includes the coding and non-coding regions both, and then these sequenced regions, they are assigned with the specific functions. So these are the two methods through which we can carry out this HGP. Now come to the process of HGP. So this HGP, it is carried out by sequencing the total genome. That means it is carried out with the help of sequence annotation method, and during this method we will study this process step by step. So during this process, the total DNA of a cell is isolated and it is then broken down into small fragments. So during sequence annotation, first the total DNA of a cell, it is isolated from the cell, and then it is broken down into small fragments with the help of nuclease enzymes. Now in next step, these DNA fragments, they are cloned by using suitable host cells and some specific vectors. So in second step, DNA fragments are cloned by using special host and specific vectors. Now the most suitable host are bacteria and yeast. And the most specific vectors for bacteria are BAC, and for yeast is YAC. Now see, both of these were asked in your board exams, that is the full form of BAC and YAC. So this BAC is bacterial artificial chromosome and YAC is yeast artificial chromosome. So in first step, the total DNA from a cell is first isolated and then it is broken down into small fragments with the help of nuclease enzyme. Then these DNA fragments, they were cloned by using some most suitable host cells and some specific vectors. These suitable hosts are bacteria and yeast, and the most specific vectors are BAC and YAC, that is bacterial artificial chromosome and each artificial chromosome. Now, in third step, these DNA fragments, they are amplified by using PCR. PCR is polymerase chain reaction. It is the process of amplification of DNA fragments. So, in next step, the DNA fragments are cloned or their copies are made by using PCR. So, this PCR resulted in amplification of DNA fragments. It resulted in amplification of DNA fragments. And this amplification of DNA fragments, it helped in sequencing of DNA fragments. Now, in next step, after amplification of DNA fragments, that is the amplified DNA fragments are now sequenced by using automated sequence technique. By using automated sequence technique, and this automated sequence technique or technology, it was developed by F. Sanger. It was developed by F. Sanger. Now, in next step, these DNA fragments during sequencing, they are sequenced from both ends. So, DNA fragments are sequenced from both ends, and they are arranged on the basis of overlapping regions. What are these overlapping regions? Suppose we are having a double stranded DNA here, that is 5' to 3' and 3' to 5'. So if an enzyme like endonuclease cuts this DNA, then it is cutting like this. So DNA is cut in this form. So this part of this segment and this part of this segment, they are known as overlapping regions. Now, so in this step, the DNA fragments are sequenced from both ends, and these sequences are arranged on the basis of overlapping regions. Now, in next step, some special computer programs were written. In order to or for alignment of DNA fragments. So in next step, some special computer programs were written for the alignment of DNA fragments. Now, these computer programs, they identified the overlapping regions and they joined them in a complete sequence or stretch. So, special computer programs, they identify the DNA stretches or the overlapping regions, and they join these stretches or DNA fragments into a single complete stretch. Now, in next step, these DNA stretches, they were assigned to different chromosomes. Now come to the most important part of this topic, that is silent features of HGP. Every year the question is asked from this part of HGP, that is silent features of HGP. So what are the silent features of this HGP? We will study one by one. So the first silent feature of this HGP was that due to HGP we came to know that human genome contains 3164.7 millions of nucleotides or the nitrogenous bases. The second silent feature of this HGP is that on an average, each gene contains 3000 nucleotides or nitrogenous bases, but this number varies greatly. And the largest known human gene, it is also an important question, the largest known human gene is dystrophin. And this dystrophin, it contains near about 2.4 millions of nucleotides. 2.4 millions of nucleotides. So on an average, each gene contains near about 3000 nucleotides, but the number varies greatly, as the largest known human gene, that is dystrophin, it contains 2.4 millions of nucleotides. Now the third silent feature of this HGP is that human genome, I am writing the short form now, human genome contains near about 30,000 genes, which is much less than earlier estimates of 40,000, or even it is 80,000, 80,000 to 1,40,000. Earlier we thought that the genes, total number of genes which are present in our body is 80,000 to 1,40,000, but due to HGP we came to know that we contains only 30,000 genes. Now the fourth silent feature of this HGP is that 99.9% of nucleotide sequences are exactly similar in all humans. So the difference occurs only in 0.1%. That is 99.9% of nucleotide sequences are exactly similar in all human beings. Further, the fifth silent feature of this HGP is only 2% of genome. Only 2% of genome present in human body codes for proteins. Next, near about only 50% of genes we have known to come the function of these genes. So the 50% of genes, their function is still today unknown. So the function of 50% of genes is still unknown. Now the seventh silent feature of our HGP is that repeated sequences forms a large portion of human genome. So repeated sequences forms a large portion of human genome. Now what are these repeated sequences? These repeated sequences are stretches of DNA sequences, which are repeated 10 to 1000 times in our genome. 10 to 1000 times means, suppose there is a sequence ATC, then again it is repeated as ATC, again it is repeated as ATC. So these repeated sequences of DNA, they forms a large portion in human genome. Further, the chromosome number 1, it have highest number of genes. Highest number of genes. That is, it contains a total of 2968 genes. So the highest number of genes are present in chromosome number 1, and chromosome Y, it have least number of genes. Least number of genes, that is, it contains only 231 genes. Further, the scientists have found near about 1.4 millions of locations in human genome. So scientists have found near about 1.4 millions of locations in our genome where single base DNA difference occurs. And this single base DNA difference, it is known as single nucleotide polymorphism. Single nucleotide polymorphism. The single nucleotide polymorphism, that is SNPs. So in our DNA, scientists have found out near about 1.4 millions of locations where only the difference of single nitrogenous base occurs, and this single difference of single nitrogenous base, it is known as single nucleotide polymorphism. Now in next topic we will be studying about the we have already studied the silent features of the ARGP. Now we will study the application of ARGP. So this ARGP, it enhances our basic knowledge about human genetics. So it enhances our basic knowledge about human genetics. It also reveals the genes which are responsible for genetic disorders such as the cancer, the Alzheimer disease, it may be, and all these. Now the third is this ARGP, it helps us in making genetically modified diets. It also provides information about hereditary disorders. Now further, this ARGP, application of ARGP also includes the treatment of genetic disorders through gene therapy. Now this ARGP, it also provides genetic identity to human beings or to humans. So these are the applications of HGP. Thank you, students.